Hi, my name is Aishi Jang, and today I'll be covering reception and cell signaling. Now here we have a signaling molecule and a receptor. And as you can see, this interaction is very specific. The signaling molecule will only bind a specific receptor. It won't just bind any old receptor. In fact, the signaling molecule is often referred to as a ligand. It is a ligand. The definition of ligand is a molecule that binds specifically to another molecule. And usually that other molecule will be much bigger. Now let's talk location of reception. We have plasma membrane receptors and intracellular receptors. For plasma membrane receptors, the ligand is a water-soluble molecule. For intracellular receptors, well, these can be either in the cytoplasm or in the nucleus. The ligand must bypass the plasma membrane and sometimes has to bypass the nuclear membrane on its own. So it must be either hydrophobic or not hydrophobic, but extremely small. So examples of hydrophobic ligands would be steroids and thyroid hormones. And an example of a very small ligand would be nitric oxide. Now there are three main types of transmembrane receptors. We have G protein-coupled receptors, receptor tyrosine kinases, and ion channel receptors. We'll be covering each of those in detail. Let's start off with G protein-coupled receptors. Each G protein-coupled receptor has 7 transmembrane alpha helices, as shown here. And they work with help from a G protein, which binds GTP. And there are many different types of G proteins that exist, and they'll do different jobs, activate different cellular signals. Now let's look at how a G protein-coupled receptor would actually work. Here we have everything in the rest state. So the G protein is inactive, and you can see this because it has GDP bound to it, not GTP. When the ligand comes in and binds to the G protein-coupled receptor, the G protein is also able to bind. And when this happens, GTP comes in and displaces GDP. So GDP gets kicked out, and this activates the G protein because it now has GTP. It's in the activated form. The active G protein with the GTP bound will dissociate from the G protein-coupled receptor, and it can then go and bind a specific enzyme, activating it, which would lead to a specific cellular response. The ligands will bind, dissociate. It depends on the concentration of the ligand in the environment. So at a given time, there'll be a certain number of G protein-coupled receptors that have a ligand bound to it, and then there will be those that do not have a ligand bound to it. When there is a ligand bound to it, the G protein-coupled receptor can go and activate other G proteins, but when the ligand leaves, like in this case, the G protein-coupled receptor is no longer able to do so. Now, G proteins are actually GTPases. They hydrolyze GTP to GDP. And that's how the G protein becomes inactive again. The GTP is hydrolyzed to GDP, and the inactive G protein dissociates from the enzyme. And so both of these guys are now inactive. And now we have a return to the initial state, and the G protein is ready to be activated again by a G protein-coupled receptor that has a ligand bound to it. Next, we'll be talking about receptor tyrosine kinases. These are the second main class of plasma membrane receptors that we will be covering. Now, they're quite different from G protein-coupled receptors. They themselves have enzymatic activity. They're kinases. And what kinases do is that they catalyze the transfer of phosphate groups. For RTKs, receptor tyrosine kinases, it's the cytoplasmic portion that actually functions to transfer the phosphate from ATP to tyrosine on the substrate protein. And another major difference is that one receptor tyrosine kinase complex can activate many transduction pathways, unlike G-coupled protein receptors where only one transduction pathway is activated. Let's go through how a receptor tyrosine kinase will actually work. Here we have an unbound ligand, and we have two inactive monomers here. So as you can see, these are tyrosine residues that will then be phosphorylated. And the portion that spans the plasma membrane is actually made up of an alpha helix. Now, when the ligand binds to each of the monomers, these two monomers will then move closer together and they will dimerize. Voila. Now, dimerization actually activates the tyrosine kinase region of each monomer. So each tyrosine kinase adds a phosphate from an ATP molecule to the tyrosine on the tail of the other monomer. Now, when phosphate groups are added to each of the tyrosine residues, now the complex is fully activated. Now that the receptor tyrosine kinase is fully activated, specific relay proteins inside the cell can then bind to the phosphorylated tyrosine residues, and each of these can induce a specific cellular response.